Hi, so in this video we're going to try and understand how to determine whether LTP has a pre or postsynaptic mechanism. And the truth is that it's probably both a pre and postsynaptic phenomenon. However, we generally don't, for the purpose of this video, we're just going to assume that it's one or the other, just to try and understand how some of the electrophysiological data in some of these experiments may lead one to understand whether LTP is pre or postsynaptic. And it's important to remember that a whole course could really be taught about this subject. It's a big debate in neuroscience, so this video is inherently going to be incomplete and even simplified. But just to understand what kind of things we're looking at here, we could draw one presynaptic neuron and one postsynaptic neuron, and this will all be done somewhere where we know where LTP has been known to take place, so we're not wasting our time. And in these experiments, we will be stimulating this presynaptic neuron, and we will be recording from this postsynaptic neuron. And the basic series of steps that we go through in these experiments are to first stimulate the presynaptic neuron and record from the post, right immediately after that stimulation. And we would do this many times, so we'd get many data points to get a good sample. And then we would induce LTP. And this is done with usually just a high frequency stimulation in the presynaptic cell. It could also be done with a Hebbian way, but in a Hebbian fashion, but usually this is the way it's done. We just have a high frequency burst sustained for a good amount of time, and then we repeat step 1. And after you repeat step 1, you can try and understand the difference between before and after LTP. So let's see an example of what one of these data points would look like in either case. So we would be recording from the postsynaptic cell. In both steps 1 and 3, we would be recording from the postsynaptic cell. And here would be one sample recording. We would have the voltage of the cell, and this would be time. And in one case, we may have just no response and maintenance of resting potential, and we'd call this a failure. And it's important to remember that in these central synapses, we do often get a lot of failures, and the probabilities of release are way lower than many would expect. And then in another case, again, this would be another postsynaptic recording. They're all postsynaptic recordings. And this time we would see a EPSP. We would actually have a response, and say that the amplitude in this case was 2 millivolts. So what we're going to be recording and keeping for our frequency histograms that we're about to get to is this amplitude change. So the amplitude of the EPSP. So this would be a 2 millivolt EPSP, or excitatory postsynaptic potential. And what we do is we take that data and we put it on a frequency histogram. And first we're going to make the histogram for before we induce LTP. So this is the data points come from step 1. And we'd have a 0 millivolt, and I'm just making these numbers up for simplicity, and maybe we'd have a lot of failures before LTP. And we'd have some successes at 2 millivolts, even fewer successes at 4, and none at 6. And then we would induce LTP to find a new, to really generate a new frequency histogram. Oh, and this axis was the amplitude of the EPSP in volts, and this was the frequency of those events. So we're literally measuring how many times each of these different amplitude EPSPs occurred, because we had many data points. So again, this would be frequency, and this would be the amplitude in volts. And now we would see, after LTP, presumably we would see a smaller amount of failures, a larger amount of 2 millivolt responses, more 4 millivolt responses, and maybe even some 6 millivolt responses. So right after we saw that the number of failures decreased, we've already seen that the distribution of this frequency histogram has changed, and we can overall see that that this distribution has changed, because we actually see an increase in the number of successes and the way they're distributed here. So what does that mean in terms of a pre or post synaptic explanation? that means that we see the first thing to analyze here is a pattern. The pattern is the increased number of successes maintain this whole number multiple of 2. So the EPSPs are all whole number multiples of 2. This first miniature EPSP here, this 2, the smallest one we have, seems to be maintained. So we have 2, 4, 6. We don't have any 3 millivolt or 5 millivolt EPSPs here. So what does that mean when we actually look at the synaptic connection? And I'm going to try and draw the synaptic connection here. And it's, if this is the presynaptic bouton and we had many synaptic vesicles with neurotransmitters in each one, and we're going to assume that that there's an equal number of neurotransmitters in each vesicle, and if the vesicles are fused, then they fuse fully in an all or none fashion. And that's actually not always the case, but for simplicity for now, we're going to assume that. And here's the the postsynaptic membrane with its receptors embedded in it. And if one of these vesicles were to fuse, it would release its contents into the cleft, and some of these neurotransmitters would actually make their way all the way across and hit the receptor. So what does it mean that we're seeing whole number multiples of 2 here? That probably means that's really reflective of this all or none release. So the fact that you either have a whole vesicle get released or no vesicle get released. And that probably means, since we're seeing more successes in these whole numbered multiples, it most likely just means that what we're seeing is a higher probability of release. And the way that happens is really you could have more vesicles, or in this presynaptic bouton, you could have a easier mechanism of fusion so that the presynaptic membrane can recruit these vesicles more easily. And the last thing is you may actually have more points of synaptic contact so that you have a higher chance that these vesicles get released. But the important thing to remember is that these vesicles are being released in an all or none fashion, and more of them are being released, which accounts for the smaller number of failures, the higher number of success rate, and the fact that the successes come in the form of whole number multiples of 2. And I say 2, it's really just the smallest EPSP we had on in the case before LTP. So, and this was after LTP. So that's what. So this leads to a presynaptic case, because we just have the number of vesicles being released changing, or the number of contacts changing, and this is really the only way that a postsynaptic cell could bring this change about would be with some type of retrograde signal. But excluding that possibility, this data would point to a presynaptic case. Now, the postsynaptic case is something we're going to get to in part 2. So postsynaptic case will be in part 2. And there we might actually get into some quantum theory that tries to put some mathematical variables to some of these ideas. And we might even get to a case that shows how the vesicle may actually not fuse fully and not be an all or none, release. We may not demonstrate an all or none release. And that's going to be a complicating factor in analyzing this data. But here we were just trying to look at one presynaptic case and the data that comes with it, and we're going to get to the postsynaptic case in part 2. And I thank you for listening, and if you're interested in this at all, you should check out part 2 and see the postsynaptic case. Thank you.